In this video, we're going to talk about why is oxygen and sugar so important to life. So as you know, if people don't breathe, they die. If people don't eat, they die. So why is oxygen and sugar so important to life? So first, we're going to look at the cell as an overall picture. Inside of the cell, there's different organelles. For example, there's the cytoplasm, that's the jelly fluid that keeps all the organelles inside. But there's also the mitochondria inside of our cells. And the mitochondria does a process called cellular respiration to make energy. So how exactly does the mitochondria do cellular respiration to make energy? Well, one of the first processes that can happen is glycolysis. When you eat sugar, sugar can diffuse from your blood into your cell, and then inside the cell's cytoplasm, glycolysis can happen where the sugar is broken down and some energy can come out of it. But the energy that you make from glycolysis is very little. What's left over after this reaction happens is a molecule called pyruvate. And pyruvate is then going to be broken down further inside of the mitochondria to make much more energy. All cells can do glycolysis because, as you can see, glycolysis happens outside of the mitochondria. You don't need a mitochondria to do glycolysis. You'll also see that the glycolysis, it also helps the NAD plus become an NADH molecule. And this NADH molecule is going to be used inside the mitochondria later on. And we'll explore that further. Okay, so mitochondria, your cell has more than one. Your muscle cells will obviously have more mitochondria because they need the most energy. There's three processes that happen inside of the mitochondria. And we're not going to go into the details. We're just going to understand the overview of why each process is important. So the first step is pyruvate oxidation. That's the first step inside the mitochondria. So that pyruvate is then broken down further to some other molecule called acetyl-CoA. We also produce carbon dioxide, waste, and we also produce more of this NADH molecule. 6, Krebs cycle. So the acetyl-CoA is used and broken down further. Okay, so when it gets further broken down, it makes NADH again, and it makes FADH2. What's FADH2 used for? We'll talk about that in a second, because NADH and FADH2, all of this is then used in the final part of the cellular respiration process that happens inside the mitochondria, in a process called oxidative phosphorylation. So we're going to zoom into what's inside of the mitochondria to see what exactly is oxidative phosphorylation. In summary so far, we just talked about how the sugar was used. We didn't talk about how the oxygen was used yet. Okay, and glycolysis broke down the sugar, and these three steps, right? Pyruvate oxidation, Krebs cycle, oxidative phosphorylation, it just helped further break down those molecules that the sugar kept breaking down into. All right, so we're going to zoom into the mitochondria to see step 7 now, the 7th vocabulary word. Okay, so inside the mitochondria, this is just one mitochondria. We have an outer mitochondrial membrane and we have an inner mitochondrial membrane. The mitochondria has two membranes. So what happens first is those NADH and the FADH2, they were holding the hydrogen, because if you look at it from before, right, the NAD plus, it gained the hydrogen as a result of the sugar breaking down. And same thing, as a result of the sugar breaking down, you were able to form FADH2. So it stored hydrogens, but more importantly, what comes with the hydrogen? An electron as well. So we're going to release those electrons back out because electron is energy, right? So the NADH, when it releases the electron, it also releases the H+. What causes the electron to be released? Some proteins that's on the mitochondrial membrane that we don't need to know the names of. So the electron gets distributed to those proteins, and those proteins then eventually distribute the electrons to oxygen. And that's where the oxygen comes in. It helps receive those electrons. So electron transport chain, the whole purpose of this is to release the electrons, but also release a bunch of H+. Now you see all the H+ is between the two membranes. Why do we need so much H+? Well, all of the energy from cellular respiration is basically made in this step So electron transport chain happened first to make all of these H pluses. These H pluses are going to then diffuse back into the mitochondria. This process is called chemiosmosis. So when the H pluses diffuse back in, the movement of them diffusing back in is going to provide the energy needed for the ADP to turn back into ATP. So the phosphate group can attach back on to allow ATP to be formed, right? And the ATP synthase is an enzyme that helps form the ATP using the energy of the H pluses diffusing through the ATP synthase. Okay, this is known as facilitated diffusion because this ATP synthase is helping the diffusion of the H pluses, right? No energy is needed because it's diffusion. Now, where do those H pluses go in the end? Well, the H pluses then attach to this oxygen, right, that had the negative charge from all those electrons being passed to it. And so there's two oxygen molecules here, right? So you're going to make two H2O as the H pluses attach onto the oxygen. And that H2O is the waste product. And that's it. So why is sugar important? Sugar is broken down in order to supply the electrons and the H pluses, right? Why is the oxygen important? Oxygen receives the electrons, right? And that way the H pluses can break apart from the NADH and the FADH2, and the H pluses, when they diffuse back in, they help form the ATP. And this is where most of the ATP is made. So glycolysis makes very little ATP. Most of the ATP is actually made right here.